Sometimes we want to make copies of all of a large amount of DNA, but in small pieces, so that we'll be able to study it in more manageable quantities later. So producing this kind of set of DNA is called a DNA library. So let's take a look at how this works. So we're taking a large amount of DNA. It could even be an entire genome from the cell of an organism, or maybe it's just a particular chromosome that we're looking at. We've got to first start with a lot of copies of the DNA. We can use a PCR to come up with that. And then we cut the DNA with multiple restriction enzymes. Since the restriction enzymes use short sequences that occur many times in many organisms' DNA, that means a lot of cuts with really small fragments. But we can expose the DNA only to the restriction enzymes for a short period of time. So that means that the restriction enzymes are not going to make it around to all of the places that they could possibly cut. So that's going to give us fragments of different sizes, some long and some short, since different ones will have different numbers of cuts. We then take our vector, whether it's a bacterial plasmid or a cosmid or whatever we've chosen, insert these fragments into the cloning vector, and transfer them into bacterial cells. Okay, we can then plate the clones and find lots of different types of combinations of genes. So when we're looking for a particular gene, some of the cells plated are going to have the gene of interest, some will not, and some might have fragments of it. So we've got to use the techniques that we've talked about to look for the right types of cells. We do this with enough vectors and produce a high enough quantity of bacteria, we can be reasonably sure of copying most of the entire genome of the organism that we're working with, or most of whatever piece of DNA we are interested in. Notice how the DNA cut by these restriction enzymes produces fragments of overlapping content. So the library is really a way of storing DNA pieces in manageable segments that we can use to construct later, or that we can use to sequence or study later. Just as we would in other cloning methods, we can look for the cells that have taken up a particular gene using a probe, or we can simply look for the protein produced. If we know what a particular gene does, sometimes we can find the appropriate protein using an antibody that matches the shape of that protein. Okay, that'll allow us to identify and plate the particular cells that we're particularly interested in. Positional cloning is another useful genetic technique. It's the process of isolating genes based on their position on a genetic map. So in this approach, we're combining the mapping techniques that we talked about in the chapter on linkage. And we're trying to find the approximate position of a particular gene based on the other genes that it's close to. This can help us narrow down where to be looking for a particular gene, and it can help us get to our molecular analysis a lot faster. So when you did all those linkage problems and you were wondering, why do we have to do this? It really is useful. Sometimes it'll help cut to the chase and help us find information out about genetic diseases a lot faster than it would if we didn't have this tool. Sometimes it's important to know the exact DNA sequence for a particular gene. Knowing this can help us to distinguish between individuals, do things like identify the correct identity of a person who's committed a crime, do a paternity test, or even to figure out whether species or populations that look very similar have diverged enough to be called separate species. It's also sometimes important to determine what the DNA sequence is for a particular gene so that we can identify the correct protein and try to figure out what it does and where the problem is if that particular gene codes for a particular genetic defect. There are lots of reasons why we might need to know a DNA sequence. So if we have our gene of interest in a DNA library, we've created a lot of copies using a vector or using a PCR. We then have a couple of choices of what to do. So the sequencing method that's been around the longest is called dideoxy sequencing. Okay, it's also known as the Sanger method. So in order to figure out how this works, let's look at a picture of a nucleotide base. The base on the left is a typical base. Okay, we have the phosphate backbone of the nucleotide. We've got our sugar. And then we have, where it says base, we have either adenine, thymine, guanine, or cytosine attached. Notice the OH group highlighted in gray at the bottom. Dideoxy sequencing involves the use of modified bases. So in these bases, the hydroxyl group has been removed and only a single hydrogen atom is present in that place. When one of these bases is incorporated into a growing chain of DNA, replication stops. Let's look at how this is used in sequencing. So in dideoxy sequencing, we use a medium that's had special bases with the OH groups removed so that DNA sequencing will be terminated at different points in the replication process. So we need to divide our DNA sample into containers in which dideoxy nucleotides for each of the 4 bases are included separately. Okay? Don't worry too much about the fact that there are so many different steps here, but I do want you to get the overall idea about what's happening here. So in each container, we have regular bases and the dideoxy bases with the missing hydroxyl group mixed in together. So we're going to use our typical enzymes that would allow us to copy the DNA, but randomly, each piece of DNA stops at a different length. Okay? In our test tube with the dideoxyadenine, we have different sizes of fragments, all which end in adenine. The same thing for our container with cytosine and guanine and thymine. So notice how each of these pieces is a different length. Of course, this is a lot easier when we're already starting with a small piece of DNA. So that's why we do things like cut with restriction enzymes. We then take a gel, and each lane is used for a different container that contains a different dideoxide base. We can then compare the length of each fragment each piece of DNA that is one base pair longer than another is going to move slightly less far down the gel. Okay? Then we can read off the sequence of the DNA. Okay, this can be done manually, but now we have a lot of computers that animate the process and make it much faster. So this picture here shows a shortcut method in which all of the bases are put into the same container and replication of the fragments of different lengths occurs together. But the labels on each of the 4 bases are a different color. They've all got a different dye with different radioactive color. Okay? Here's what a computer printout might look like for the results. It's going to put the fragments in the correct order and read the sequence for you, which is a big help when you have long pieces of DNA with possibly thousands of fragments. Okay, so the Sanger method, or the dideoxy sequencing method, has been around a pretty long time. Okay, I think 1970 was the year in which it was first introduced. Since then, some faster methods have been introduced as well. We won't go into a lot of detail, but just be aware that these are the names of some of them: Illumina, pyrosequencing, and third-generation sequencing. Scientists keep finding better ways to animate this process and to keep different steps of the process happening at the same time so we can get our data faster. And of course, that's always the goal of scientists, is to publish quickly and get your information out there before somebody else does. Okay, DNA fingerprinting is a really useful, quick and easy way to identify individual people or individual animals or other types of organisms. Okay, it's commonly used in crime investigation when various suspects are being analyzed to see who left their DNA at a crime scene. So DNA fingerprinting involves microsatellites. Okay, microsatellites are short tandem repeats. That means that the same bases are repeated over and over again, usually in groups of 2 or 3 letters. But the length of these repetitive sequences differs depending upon individuals. So in one person, a particular sequence might be replicated 10 times in a row, and for another person, 30 times in a row. The sizes of these fragments can be determined after the DNA is amplified by PCR, and the sizes of the fragments are determined on gel electrophoresis. So for a particular locus or a particular gene that can include a tandem repeat, you'll see a tall peak with a lot of DNA for a homozygote that has 2 copies of the same number of tandem repeats. In a heterozygote, you'll see 2 shorter peaks. Let's take a look at what this looks like. Okay, so let's say that in one individual, they've got 8 repeats of this CA-CA sequence. Another individual only has 2 repeats at that particular locus. Okay, we know what sequences are flanking on either side, so we use the right type of enzyme to cut at the right particular place. Individual 2 with a shorter number of repeats is going to have a piece of DNA that moves farther down the gel. The larger fragment from the other individual won't move as far, and these individuals can be distinguished from one another. On a computer analysis printout, these are showing different peaks. So here we have an individual that's homozygous at a particular locus that's been named D8S1179. Okay, it's got two pieces of DNA that are the same size. At these other loci here in the middle, D21S11 and D7S820, the individual is heterozygous. One of the pieces of DNA on one of their chromosomes is longer than the other. The height of the peak shows the amount of DNA that's that particular fragment size. This diagram here shows the standard loci used for DNA fingerprinting. Okay, so this is often used by police detectives trying to identify suspects, and it's also used in things like paternity tests. So notice that because there are 13 different loci that we're looking at, and there's a wide range, look at these number of repeats, okay? Here's a very variable one. It's repeated anywhere from 12 to 51 times in different individuals. So that means 23 different alleles are known in the human population. That's a lot of different possibilities. Okay? If you combine all of these genes together, and you were to look at every locus, you'd find several billion different possibilities. So it's unlikely that any two people have exactly the same genotype for all 13 of these loci. Okay, so these loci are better to use than a simple gene that has only two possible variants. And it's also a lot easier to just look at the size of the fragment than it is to sequence a large portion of a person's genome. When we want to be able to use this in so many ways, we want the fingerprinting to be relatively quick and easy. So here we have two different suspects, 1 and 2, for a particular crime, and it's easy to see which one matches the sample from the crime scene. So just because we've identified a particular gene doesn't mean that we necessarily know what it does. Okay, there are a lot of phenotypes that we haven't linked to a particular gene, and there are a lot of genes that we haven't linked to a particular phenotype. So scientists use several different approaches, including forward and reverse genetics, to try and figure out which phenotypes are associated with which genotypes. So forward genetics is a general overall approach that can involve several different types of molecular techniques. But the idea behind forward genetics is that you begin with a phenotype and then you try to narrow down the gene that encodes that phenotype. Reverse genetics has the opposite approach. It begins with a gene whose function is unknown. The scientist then tries to induce mutations in that gene, or maybe they break up that gene using a restriction enzyme or using CRISPR, make that gene defective, and then compare wild-type individuals with individuals where that gene has been disrupted or removed. Okay, compare the different types of phenotype, and you'll figure out what the gene does. So let's take a moment to answer this question. Suppose you're interested in the immune function of mice, and you induce several random mutations in a number of mouse genes, and then you determine which of the resulting mice that have a particular mutation have impaired immune function. Okay, so you're interested in which genes have been affected, and they're probably involved in the immune system. Would you say that this approach is a forward genetics, a reverse genetics approach, or both or neither? Okay, hopefully you said reverse genetics because you're starting with a gene of unknown function, you're randomly inducing mutations, and then you're looking for the mutants that have that particular effect. So sometimes the goal of genetic research is to create transgenic organisms. Okay? If you've altered the DNA sequence of an organism's genome, it's known to be transgenic. Okay? And the transgene is the one that's been transferred from a different organism into the organism that carries it. Okay? So you may have also know this technique by another name, genetic engineering. You may be aware that foods in the grocery store have genes that have been introduced from different species to give them a desired characteristic. When we talked about cloning particular genes using bacteria and then plating them, those are also transgenic bacteria. Scientists often create transgenic mice because mice have been known to be a useful model organism. Since mice are mammals, they have a lot of genes in common with humans, and they reproduce very quickly, which make them a good study organism that make it easy to do fast and inexpensive research. So transgenic mice can be produced by taking a recently fertilized egg and injecting the DNA into that newly formed zygote. If the zygote is then implanted into a pregnant female, we can then determine which of the offspring have taken up the introduced gene by doing genetic testing using gel electrophoresis. Once we've got one transgenic mouse, it can take many trials to get this to work, but once we've got one transgenic mouse, we can create more by ordinary breeding. Perhaps inbreeding the mice to get a homozygous or pure breeding strain for that particular gene. Then we can do a lot of studies and experiments on how that works. Okay? We can look at how the environment affects the expression of that particular gene. So sometimes we want to use knockout mice. Let's say that we know we have a particular gene, we want to know what it does. We disrupt that gene, cutting the DNA in an appropriate place to fully disable that gene. Okay, then we look at how the mice are different from wild-type mice, and then we know what that gene does. Knock-in mice have a particular gene inserted at a specific location. Okay, so these are good for studying phenotypes that are gain-of-function mutations or produce something new and unusual in another organism. So no doubt, many of the techniques that we use as geneticists are highly controversial. Some of them have had incredibly excellent results. By cloning genes for insulin and growing them in bacteria, we've been able to easily produce products that help diabetics. We can also produce vaccines and other medical products by getting bacteria to do our work quickly and produce lots of a particular protein. We can have specialized bacteria that solve all kinds of environmental problems. Scientists are now looking into research on bacteria that digest environmental toxins like oil and like plastic that's made its way into the ocean. Scientists have found bacteria like this that have evolved on their own, and they're figuring out how to get these genes into bacteria that can grow in other places in order to do special things. Genetic engineering has increased the speed at which agricultural products like crops and livestock have been able to change. Of course, a lot of these things are very controversial. Some people are concerned about the rate of change in the food products that they're eating, and they're concerned that there's not enough safety testing before these foods end up on their plates. And in fact, we in America have products with high amounts of gluten produced by genetic engineering. In Europe, this isn't allowed. They can increase the amount of gluten through selective breeding, but the amount of gene product doesn't go up quite as fast. As a result, more people in America have developed gluten sensitivities and have had health problems from eating grains with too much gluten in them. Other people say that genetic engineering is awesome. Some genetic engineering has created pesticide-resistant crops, so we have to spray our crops less often, and there's less risk of getting pesticide residue on the food that we eat. So obviously there can be a lot of positive and negative effects of all of these technologies. In medical science, we can now test people more easily for particular disorders. Many disorders can be treated more easily with early diagnosis. One example is the gene for PKU, or phenylketonuria. It's a disease that if a baby is tested for it at birth, it can be treated with a special diet. But if the diet isn't implemented immediately when the baby's very young, the baby can suffer some brain damage. So obviously this is a test you really want to have done. And it's routinely done in all hospitals all over the United States. Gene therapy is the idea that we are replacing defective genes with fully functional genes in curing people of genetic diseases. Although a lot of early trials in gene therapy didn't work very well, we're now finding some more promising methods, like the CRISPR-Cas9 technique, that are allowing this process to work more efficiently and more effectively. And maybe someday this will be a reality.